Hello, my name is Cy Gart. Welcome to this video about an argument for God from biology. There are many arguments or pointers to God that come from science, mostly from physics. We know about the cosmological argument. We know about the fine-tuning argument. There are some other arguments, or at least pointers, from physics that point to the existence of something beyond the natural world. Some come from quantum mechanics, from the observer effect, and there are some other scientific arguments that suggest that a God could be real. But one area of science which seems deficient in such arguments or pointers is biology. In fact, it's been found that very few biologists are theists compared to the other sciences. One reason for that is that while life is beautifully complex, ornate, appears to be designed, the reason that biology does not appear often in any discussion of the scientific case for God is that all of that is often explained by natural selection and biological evolution. And that explanation is not bad. Many of the beautiful designs that we see in living organisms do in fact originate from the natural process of biological evolution. But what has not been looked at is the question of evolution itself. In this talk, I am going to present an argument from biology which is focused on some of the aspects of evolution that are not usually considered. The argument consists of 5 propositions. The first one is that evolution is real. Some form of evolution by natural selection, ENS, is certainly possible. And ENS can explain at least some of the diversity of life, if not all of it. Now, when I say it's possible, I'm talking about various ways that people define evolution. That can include microevolution of plants and animals, the evolution of bacteria that survive antibiotics, and evolution within species and even families. And that kind of evolution is accepted by practically everyone, including many young Earth creationists. And the first proposition is that such evolution by natural selection, even if it does not explain every single aspect of life, certainly does explain some of it and some of the diversity and the changes that we see in life. I don't think that any argument could be made scientifically that this proposition is false. Here we see the typical phylogenetic tree that illustrates how evolution works. You would find trees like this in any biological textbook or any book describing evolution. But there's something interesting about this tree, which is that it comes from a young Earth creationist site, Answers in Genesis, and what it shows is the kind of evolution that creationists believe is correct, that explains how it is that animals from the ark, after the flood was over, were able to populate the earth. This takes into account the fact that not every single species alive today could have possibly fit on the ark. Therefore, the theory is that certain animals who were representative of many other modern animals were on the ark, and they evolved into the current group of animals that we see today. So if we look at the main cat kind, the branch on the right, we can see that one particular cat kind came on the ark, and there is a statue of that cat, ancestor of all modern cats, in the Ark Museum, and it doesn't look like any cat we know of today. It looks like an amalgam of all the cats. And with time, going up the tree, we see that there are branches, just as regular biological evolutionists propose. And these branches eventually led to all the modern cats, such as panthers and big cats and the smaller cats, domestic cats. Cheetahs, lions, tigers, they're all here, and they all evolved from the original cat kind. Now, while this doesn't really fit with a standard model of biological evolution, it is a form of evolution that occurs by natural selection. So when I said that this first proposition is not argued against by anyone, I'm including people who reject the general theory of biological evolution to explain all of how living creatures came to be and the diversity of modern life, but it still uses the standard principles of natural selection and everything that occurs in biological evolution. So again, there are really no arguments against the idea that, at a minimum, evolution by natural selection explains the diversity of life to some extent. And that's exactly as far as that first proposition goes. The second proposition is that evolution by natural selection requires high accuracy self-replication, which I'm abbreviating HASR, of living cells. Now, this is a fact that is well known in biology, but it is often left out of discussions of the evolutionary process or the mechanism, which is a shame because it's absolutely critical. The only way evolution can work is if the cells that have a selective advantage replicate that advantage to their descendants. And when I say high accuracy, I'm meaning much greater than 99% accurate. What that means is that cell replicates must be identical to each other and the parent cell in at least 99% of their properties and components. At any lower accuracy, what we find is an error catastrophe. In other words, the mistakes, the errors that come up if the accuracy of replication is poor can lead to death. Or, if not death, they could lead to a failure to inherit the beneficial mutations that lead to eventually a selective advantage and natural selection. This again is something that is not disputable. It's a biological fact that professional biologists, especially those working in the origin of life, are very aware of. The problem is that it's often not stressed enough when we talk about the biology of evolution or the evolution of biology. This fact, this proposition number 2, forms the central core, the central basis of the argument that I'm going to make. It's not just that cells must replicate themselves, but they have to replicate themselves with a very high degree of accuracy. So here's an example of a cell which has divided into 2 cells, and what we see is that these 2 cells have to be equal to each other. In modern life, 99.9999% is roughly the degree of accuracy of the replication when a cell divides in 2. So this cell has split into 2, and as you see, the 2 are almost identical, totally identical from our eye. There might be tiny little variations. And when I say that the cell is identical, I mean that all the components are identical. So you have the same proteins, the same nucleic acids, the same lipids. All the molecules of the cells, the structures of the cell are or must be the same, with very small differences between them. Any variant that gave a selective advantage to the cell, as opposed to some of the other cells in its environment, could very well be lost if the replication accuracy is not close to perfect. Now, of course, it can't be perfect because there has to be some variation, or there would never be evolution either. And sometimes we talk about the error rate. The error rate is 1 minus this number. So in this case, the error rate or the mutation rate would be 0.00001%. The third proposition, which seems like something that does not need to be stated, it seems like a truism, is that all life is composed of chemicals, molecules. Many critical biochemicals, in other words, chemicals that are found in life, are polymers, long chains, and these are usually not found outside of life. They include such things as proteins, nucleic acids, and lipids. The monomeric units of these polymers which are amino acids for the proteins, nucleotides for the nucleic acids, and fats for the lipids, are found in non-living nature, but the polymers, in other words, the chains that these monomers make up to form the proteins, nucleic acids, and lipids, are generally only found within life. There are many other molecules in living organisms that are not polymers. They include the monomers that we see here, and they also include everything from sodium chloride to all sorts of inorganic ions and other materials, a vast variety. There are also structures within cells, such as membranes, which are also composed of molecules. When we talk about the components of living cells, we are speaking of chemicals, which follow the laws of chemistry. This shows some of the variety of chemicals that we find in living cells. The right panels show nucleic acids. The next panel down, the middle panel, shows 4 amino acids as part of a protein. If chemistry is not your thing, that's fine. I'm just showing this for illustrative purposes. The third panel down, the bottom one, is the structure of one of the phospholipids that make up membranes. The larger panel on the left shows a collection of various organic molecules. And of course, what this slide does not show are the large number of inorganic molecules. The major molecule that's everywhere in living cells is water, H2O. The fourth proposition is that no physical entity outside of life is capable of high accuracy self-replication. This is the most controversial of the propositions. Now, DNA replicates in life, of course, but it requires many enzymes for it to happen, and if you just leave DNA in the natural world, it does not self-replicate. And there's nothing else that self-replicates. Let's see that I see a house, say the one on the left, which I really like, and I want to replicate it. I want to build another one just for me. And I could do that with some work, but if I now said, let's replicate everything inside the house, all the books, all the pictures, all the furniture, that would be very difficult. Not impossible, but it would take a lot of time and a lot of effort. As hard as it would be for me or anybody to replicate a house and all its contents, one thing we know for sure: no house can ever replicate itself. And we can't imagine how a house could replicate itself. There really is nothing in the world outside of life that can replicate itself. This is a really important concept to understand because it is, in my mind, one of the key reasons that life is so different from everything else that we see. And that high accuracy self-replication of the cell and all its contents is a critical part of how evolution, which means all of biology, works. Now, as hard as it would be to replicate a house with all the contents, all the different books, there may be hundreds of books and the various paintings, that's nothing compared to how hard it is to replicate a cell. There are 10 billion protein molecules in the typical cell. There are almost 10 to the 12th molecules in total in a living cell. That's a huge amount of material. Obviously, these molecules don't replicate themselves, but the cells replicate themselves. And they had to figure out a way that all the contents, all the material that's within the cell, could be replicated at high accuracy. So it's no wonder that scientists studying the origin of life have been looking for self-replicating molecules, especially RNA, because it's pretty well known that DNA requires a lot of help to replicate itself. RNA, on the other hand, does have properties that make it a little bit like an enzyme, so theoretically it could replicate itself. But the search for self-replicating RNA has been very difficult. The three people mentioned here, Jack Szostak, John Sutherland, and Gerald Joyce, are the three leading researchers in the field of the origin of life. And I want to read what they say about this idea of replication or self-replication of the only molecule that really is a candidate for that, which is RNA. Sostak says, the chemical replication of oligonucleotides long enough to perform catalytic functions is particularly problematic because of the low efficiency of non-enzymatic template copying. In other words, he's saying it's difficult. The current lack of an efficient pathway for non-enzymatic RNA replication is the main obstacle towards building a model protocell capable of growth, division, and therefore Darwinian evolution. That's basically stating my point, but in much more elegant and scientific terms. John Sutherland says, the transition from the chemistry of RNA monomers to the biology of a self-replicating system has been challenged by the many problems encountered with multiple cycles of non-enzymatic RNA replication. In other words, it's very hard for non-enzymatic, meaning self-replication of RNA. He further says, further investigations will be needed to provide explanations to the many challenges in the field, including the possibility of preventing RNA strand invasion and inhibition in the course of multiple replication cycles. These are problems that these scientists and others have been facing for a long time, and they are strong challenges. And finally, Gerald Joyce says, thus, the fidelity of RNA polymerization should be considered a major impediment to the construction of a self-sustained RNA-based evolving system. The propagation of heritable information requires both efficient and accurate synthesis of genetic molecules. Again, making my point. So while these scientists are still working, and what they are all saying is this is an extremely difficult challenge to face. And of course, I should point out that's only one problem with the origin of life, but it's a serious one, and it relates specifically to my argument in this proposition that as far as we know at this moment, there are no chemicals, there are no entities outside of living cells that are capable of high accuracy self-replication. The final proposition, which is basically a statement of fact, is that even though nothing outside of life, including any molecule, is capable of high accuracy self-replication, life, meaning cells and all living organisms, is capable of high accuracy self-replication at a level of 99.9999%. Again, this is a fact which is well known and has been demonstrated for the cells of all living creatures. There's no disputing on this. What it does do is it raises some questions. So the question is, if chemicals cannot self-replicate, and self-replication is required for evolution, how did replication of the biochemical components of life begin? And how did evolution begin? I haven't told you how it is possible for cells to self-replicate all the material that's in them if these chemicals, these molecules, don't self-replicate by themselves. And the answer is there is a process whereby the DNA, which does replicate itself from one cell to the next, that DNA, based on its genetic code, is able to orchestrate the replication of all the proteins that were in the original cell. And those proteins include enzymes which themselves then act as catalysts to reproduce all the components, all the chemical components that were in the original cell. This is a very complicated process. It's called protein synthesis or translation. We know how that system works, and it's quite remarkable. It will be the subject of another video. So the conclusions are fairly straightforward. Assuming that life began as a series of complex chemical reactions that eventually evolved into a living cell is fallacious. The problem with this idea is the words eventually evolved, because it implies that evolution was present from the very beginning, whereas in fact it was not. It required a certain level of sophisticated ability to do self-replication before you could have any evolution at all. So therefore, the origin of life must have involved something in addition to natural chemical reaction in order to allow for the origin of evolution itself. What that something in addition is not clear. It could be miraculous. It could be something that actually does follow natural law as we know it, but we don't understand what that is yet. There could be a form of evolution that we know nothing about that could explain the growth of chemical complexity in cells and eventually lead to biological evolution. So at this point, we really don't know what that additional ingredient is that is needed for the beginning of evolution, which allows for the beginning of life. Now, the complex biochemical reactions of modern living systems are mostly understood. They include energy production and conversion, the heredity of genetic information through copying of the DNA, the heredity of phenotypes, which occurs through protein synthesis according to the genetic code. There's also some very complex systems such as gene regulation, how genes are turned on and off. And there's metabolic regulation to allow for the synthesis and breakdown of the components of the cell. Cells communicate with each other, and there are some sophisticated systems for that. And there are also error correction systems, which are a whole field in itself. All of these processes were present in what we call the last universal common ancestor, or LUCA. So we don't know how any of these systems evolved, because it appears as if they were all there in the very first cell we can go back to. Now, since LUCA, all of these processes have been improved and have become what we now see today, very complex, very elaborate, and absolutely beautiful. I should say that, as the title says, we understand the great part of how all of these systems work, thanks to the results of scientific research. But what we don't know is their origins. So even though we know the complex biochemical processes by which these systems work, their origins, in almost every case, remain a mystery. Thank you for watching this video about my idea for an argument from biology. It is still a work in progress, so I would really appreciate any comments, even criticisms, to help refine the arguments that are made here. So thanks again, and God bless you all.